Thank you very much, Brigitte, for agreeing to give a Build a Cell seminar, and you can get started.
Thank you very much. And it's really my pleasure to give my first talk in this seminar session. Okay, so my research group, in my research group, we are fascinated with the liver. So the liver is not only the chemistry factory in our body, but it's also the second most transplanted organ in the world. So when we think about it, every year in Europe, the same amount of patients receive a transplant, while the same around 1,000 people still remain on the list, waiting for an organ transplant. And approximately 10% of those patients, unfortunately, pass without ever having received an organ. Now, tissue engineering or regenerative medicine is certainly trying to fill this gap by ex vivo growing tissue and lowering the pressure on organ donation. However, if you think about the liver, so the liver is an organ that weighs approximately 1.5 kilos. And if we think about in number of cells, this is an incredible amount of cells. We need to grow ex vivo to be then transplanted, even if we consider that we only need approximately 20% of a liver for a patient to survive. Now we have to have these cells from somewhere, and we have to expand them and preserve their phenotype. Today, we are just not as advanced as we could be in tissue engineering, that the cell source is not a problem yet. But at some point, we will have advanced, and then we hopefully will not face the same problem with getting enough mammalian cells to actually do tissue engineering as we today have with organ transplants. So what my research group is trying to do is to mitigate this kind of challenge. So we are not trying to grow a synthetic liver in our lab and then to transplant that. But what we are wanting to do is we are trying to design artificial cells that can act as supporters for living mammalian cells. So probably in a ratio 8:2, so 20% synthetic cells and 80% mammalian cells. And even that would mean billions of cells we don't need to grow or from natural sources. And so my research group is doing this across the length scale. So we're trying to synthesize small organic molecules, typically artificial enzymes that can take over certain function. We then assemble this into nano-sized organelles with the aim to have nanoreactors that imitate certain functions of our living cells, which we then eventually assemble. So they are then micro-sized objects. And finally, we try to integrate that with living mammalian cells into semi-synthetic tissue. Now, this integration we can do in very different ways. We can have either sort of one-to-one integration, one mammalian cell interacting with one artificial cell. We can do it on small scale, sort of millimeter size that aggregates of artificial and mammalian cells form, or then with the advancement of 3D bioprinting, we now can make or aim to make the semi-synthetic tissue in the centimeter scale. Now, there are obviously two aspects to this. There's on one hand, what kind of artificial cell are we using? And on the other hand, how are we going to integrate them with the living mammalian cells? So first, I would like to show you some of our recent efforts we did when it comes to assembling our artificial cells. So we have been focusing a lot of our efforts on basing our artificial cells on polymer lipid hybrid vesicles. So these are vesicles that are assembled from amphiphilic block copolymer together with lipids, with the aim to create a hybrid membrane that preserves the ability of lipids for self-assembly while offering all the opportunities modern polymer chemistry has. So over the past 8-10 years, we have done a lot of basic physical-chemical assessment of how these membranes assemble, how they partition, how that affects the permeability or activity of enzymes when they are kept encapsulated or associated with the membrane. And so what makes our hybrid vesicles different from other efforts in the field is that we decided from the beginning that our hydrophobic blocks need to be made out of something that perfectly integrates with the lipids, such that we don't have phase separation within the membrane. And what we chose to do, and what we chose for that purpose, is that our hydrophobic block is essentially a polymerized sterol, mostly cholesterol. And so we designed a variety of amphiphilic block copolymers, all having cholesterol-pendant groups in the hydrophobic block, and then a diverse variety of hydrophilic extension, for instance, polycarboxyethyl acrylate in the example here, which we then can assemble into giant or small hybrid vesicles. And so recently, we had a very interesting collaboration with the young faculty here in Aarhus University, Joseph Lyons, whose favorite enzyme is the flipase. So the flipase is a transmembrane enzyme that flips the lipids between the two leaflets. And the flipase Joe is working with is specific for DOPS lipids. So while we originally wanted to investigate if we can use this lipid to change the transmembrane asymmetry in our hybrid vesicle post-assembly, we actually came by a much more interesting aspect. But before that, I would like to convince you that we actually can reconstitute this flipase into our hybrid vesicles. So first of all, it's an ATP-dependent enzyme, so we can measure how the ATP is depleted in the system. But how do we actually prove that we are flipping the lipids within those systems? And so the assay we're using here is shown that we reconstitute the enzyme, we add ATP, we let the reaction go so that the DOPS is flipped. And then we use annexine, which is a calcium-mediated protein that specifically binds to DOPS and cannot cross our membrane. So essentially, by monitoring the fluorescence intensity that comes from our vesicles, we monitor how much annexine is binding to the outer leaflets, and with that, how much DOPS is actually present in the outer membrane of our leaflets. So I should also mention that the flipase luckily has a very interesting geometry, such that it favors the small part. So the ATP hydrolyzing part is the bigger one on the outside, and it inserts in this orientation with almost 70% from all the enzymes. So what we can see then when we do confocal laser scanning microscopy, and in orange here is the protein, so annexine, that binds to DOPS, that when we don't have the enzyme present, we do have obviously some binding because we have DOPS in the membrane, but it doesn't matter if we add ATP or not. Then when we have the flipase reconstituted and we don't add ATP, we have clearly a slightly brighter fluorescence signal, so meaning that during the reconstitution, we do have some lipid rearrangement. But then when we add the ATP, the fluorescent intensity goes substantially up, suggesting that we're really flipping the DOPS from the inner leaflet to the outer leaflet, which is then also here quantitatively confirmed by doing image analysis. But what was much more interesting, what Paula observed, the PhD student working on this project, is that she had a lot of snowmans in her samples. And so we made her count those snowmans, and between all the different repeats she did when she has the flipase present and adds ATP, approximately 30% of the snapshot in time she takes are snowmans. So the question is now, obviously, where does these snowmans come from and what happens to them? And what she then did, she took movies of her samples. And what you can see here in this image, if you just focus on the boxes, you can really see that those vesicles, they constrict and eventually divide. Sometimes here in the middle, you can even see that one vesicle actually splits up in multiple individual vesicles within a few minutes. So if we have a little closer look at just that one specific example, you can see here that the vesicle really constricts and then separates and moves apart from each other. So this does work for very specific membrane compositions. It doesn't work always. We never managed to reconstitute flippase into purely lipid-based vesicles. And if we change the polymer type we add to our hybrid vesicles, we can see that we don't have division, but this vesicle essentially constricts and then eventually bounces back to form back into its spherical shape. So it cannot divide. And what's the difference between these two membranes is essentially the membrane rigidity. So the first vesicle you saw, that's by far the most flexible membrane, which allows then that we can divide this vesicle using one single membrane modulating protein, which is really exciting because it's very simple. And it does not have an inherited limitation that we could not keep developing this towards an actual, rudimentary self-sustained system by making the vesicles grow and divide again if we externally add small feeder vesicles to it. So that is one aspect from our recent hybrid membrane vesicles. But if we do want to integrate our artificial cells with the mammalian cells, we cannot work with vesicles. At least in our hands, when we use giant vesicles that only have glucose in their void, the vesicles will be destroyed when the mammalian cells start to interact or attach to them. So one way we're trying to go around this is by integrating cytoskeleton components into the lumen of our giant vesicles. And so one example I would like to share with you was a collaboration with Professor Alan Liu's lab from Michigan. So Paula went to visit him last year for a few months. And so we essentially decided that we could probably combine our expertise, his expertise on microtubule polymerization and our hybrid vesicles. And then we wanted to see, can we actually change what kind of microtubule network we're obtaining depending on how much amphiphilic block copolymer we're adding to our hybrid vesicles, and how does that develop over time. So when Paula did those experiments and took microscope images, that's what she saw. So it's a very, very mixed population of a lot of different things, which meant that we either could have sorted, but what we decided is to just classify the different vesicles. So in each population, we would have empty vesicles, vesicles that have just fluorescent tubulin, vesicles that form a network, vesicles that had everything associated to the membrane, and vesicles that had like these collapsed aggregated microtubules. And what then was particularly interesting, so what we wanted to have is number 3 here in the middle. So we wanted to have that kind of network structure. And when we had a closer look at that population, we actually noticed that they are doing quite some interesting membrane deformation, that they had quite some interesting membrane deformations within that group. So now the question is, what happens if we now take our hybrid vesicles with a relatively low polymer content, we let the microtubules polymerize and we wait. And then we count how many vesicles are in which group. And when we have this low polymer content, it's a relatively mixed population. After 1 hour, group number 3, so the network we wanted to have, is dominating. But if we wait for 24 hours, it's an even distribution between all the different options we had. And what was particularly interesting, if we compared the ones that were remained as a network and the microtubules that were attached to the membrane, it was clearly size dependent. So the smaller vesicles had higher amounts of membrane-associated microtubules compared to the larger ones where the network was preserved. But what was then the most interesting aspect here is the fact that when we now increase the polymer content in our vesicles, And we again do the same experiment. We check after 1 hour and 24 hours. We can see that in both cases, the group number 3, so the network, is preserved. So suggesting that the presence of the polymer really stable in the membrane really stabilized the network, even prevented it from collapsing on itself and aggregating. So microtubules are obviously one component only in the cytoskeleton, and the other one is actin. So we have some efforts ongoing in my group looking at actin polymerization, but this is the project from one of my postdocs, but it had a little different motivation. So there is a Listeria bacteria. So it's a bacteria that is very clever, and when it's within our mammalian cell, it essentially uses the machinery of our cells to create an actin, to create actin filaments on its surface, such that it can move. It can even move to leave the cells and then infect neighboring cells. So this mechanism is so powerful that Listeria is really very good in crossing our intestinal barrier, or sometimes even the blood-brain barrier. So we were wondering if we can actually imitate that in a synthetic system. So can we actually make a particle move with the same mechanism, or a simplified mechanism of this Listeria bacteria? And can we then do this inside of a vesicle to essentially form a cytoskeleton? And so this is an effort that is not purely rooted in our artificial cell effort, but it's more like part of our active meta direction in my group. So we're looking at micro and nanomotors. So these are particles that can convert any kind of energy into kinetic energy such that they're outperforming Brownian motion. And these actin polymerizing swimmers are just one of the examples in this area. So what we ended up doing is we assembling, so we had ACTA, so the actin polymerizing protein, we had that expressed. We immobilizing it onto the surface of a particle, and then we using cell lysate from HUVEC cells such that we have the entire component we need from the cells to actually have this polymerization happening. And so the mechanism why this bacteria is moving, or our particle is moving, is the fact that when we add one monomer unit to this growing filament, that creates a force which pushes the particle forward. So essentially, it turns the random Brownian motion, because the insertion of this monomer prevents the particle from moving backwards, which means that the thermal energy is turned into somewhat directional motion, and that makes the particle move. So not going into details here, but what you can see when we have the controls, so ACTA versus BSA, so actin polymerizing and just the controls, that we do have enhanced velocity of those particles below 1 square micrometer per second. So it's not very fast, but it clearly moves. And when we then encapsulate these particles together with all the cell lysate into our giant hybrid vesicles, we can really form these very beautiful 3D networks that are sort of floating within the void. So if we do the same experiments, but we do not use particles, just everything in bulk, we never saw any substantial network formation as we see here. Okay, so these were a few examples of our most recent advancement with artificial cells. Now, one other interesting aspect, though, when it comes to interacting with nature, smaller or bigger versions, it's always the question, well, how do you go inside and outside of cells or artificial cells? And so we really asked the questions for quite some time before we decided on these experiments here together with Miguel. How do we actually show that a nanoparticle can cross the membrane? Because in the actin example you've seen before, one could expect if those motors have enough power, they could technically probably escape the vesicles. So what we decided to do, though, as a first step towards that is to use magnetic swimmers, which we can pull with a controlled force and then see how they are interacting with our giant vesicles. So the way this looks is essentially we're making giant vesicles with different lipid compositions. We add our magnetic motors to it, we apply a field, and then what can happen is that the motors either just pass by the vesicles, cross into it, or get stuck in the membrane. And then when we record these movies, we can afterwards analyze the trajectories and then should be able to discriminate between motors that just move in the bulk, so they are fast. Other motors, they cross the membrane and slow down because the inside of the vesicle is higher viscosity than outside, or others will just immediately stop. Now, together with Professor André Hovaka from the University of Southampton, we identified an opportunity to actually analyze these trajectories, because we didn't just want to see an average speed, but we wanted to actually see what happens what happens with the velocity of a trajectory that comes fast from the outside, gets stuck at the membrane, and eventually crosses into it. And so what we are looking at here is we're comparing how the distance over time, so how far a particle can move within a given period of time, which is then directly correlated how fast those particles are. And when we do this with all the trajectories in a movie, we can then discriminate between particles that cross, that get stuck, or move inside or outside, while we're mostly interested in the ones that are getting stuck. And so I would just like to emphasize one highlight result from that effort. So obviously, if you think about it, or also discuss it with people or get review or comments back, people would say, well, how difficult can it be? The stronger your magnet is, the more likely your particles are going to cross into the hybrid vesicles, which is not wrong, but it's also not the entire story, because what you see here is a comparison. So the image here shows polylysin coated motors, so they have a positive charge, and the vesicle itself is sweet ionic. And then if we initially have all the motors, they're just distributed and nothing much sits inside of the vesicles. When we apply the magnet on this side, you can see that the motors are moving, crossing, and accumulate within the vesicles. And so we can then quantify how many are actually have crossed. And so it's around 13% of all the motors are able to go across the membrane. Now, if we do this exact same experiment, but the only thing we're changing is the surface coating of the motors. So we're pegylating the motors. Everything else is exactly the same. And here we do not see any crossing at all. So they all get either stuck or move just outside, away, or around of the giant vesicles, which is a very interesting aspect to keep in mind when we start to want to use our artificial cells to communicate with their environment by releasing or internalizing not just individual molecules, but probably larger sized nanoparticles. Okay, but with that, let me go back to how we're now going to integrate our artificial cells with our mammalian cells. So first of all, as soon as we start to do that, we always have to opt for simplicity over complexity, because we cannot have any single ingredient in our artificial cells, or a product or a substrate or anything we need that is cytotoxic, which is obvious. But what it comes in addition is we need structural stability and resilience, and we need that for quite a long time. So we need that over weeks or sometimes even months, depending on what kind of experiment we're doing. And since we're also wanting to use these artificial cells for 3D printing, we actually need a substantial yield that we do have enough volume or mass to do so. And in addition, what we also wanted to do when we're integrating our artificial cells with mammalian cells, we really would like to see if we can establish communication and not just have integration due to local proximity. So cellular communication in our tissues is absolutely important to ensure that this multicellular tissue gives a coordinated response to changes in the environment. So essentially, cellular signaling is a signal is being transferred, received by another cell, which then induces a response, which then normally gives a positive feedback or negative feedback loop to the sender cell. And because this is such an important aspect of life, the artificial cell community is certainly also trying to establish communicating artificial cell populations, or to establish communication between artificial cells and living cells, bacteria, or mammalian cells, for instance. So what we are focusing on, we would eventually like to have a reciprocal communication loop between an artificial cell and a mammalian cell, essentially a beneficial feedback loop. So what we want to have is a signal transfer from the artificial to the mammalian cell, then a signal processing on both ends that it turns into a beneficial loop. So what we essentially have to do, we have to teach the language of biology using the tools of chemistry to our artificial cells. Today, we cannot accomplish that, but we started with some initial steps. So first of all, almost 5 years ago, we accomplished the first step, the signal transfer step. And so what we did at that point, we decided, okay, we are interested in the liver, and we're going to go very simple by just creating a fluorescent product that we can, via simple diffusion, transfer from the artificial cells to the mammalian cells. And because we are interested in the liver, our artificial cells contain a small organic unit that imitates the P450 enzyme, which is a super enzyme family that is involved in the metabolism of endogenous and xenobiotic compounds in our liver. And with this active compound here, with this active core, looking very interesting for chemists to actually imitate. So what we initially did, we synthesized a variety of water-soluble metalloporphyrin and tested them if they can actually be catalytically active at 37 degrees in an environment that will make a liver cell survive. So I should mention that these metalloporphyrins have been widely used in organic synthesis, but very often in organic solvent at too high temperatures, which was for our purpose not useful. So you can see here just two of our favorite options, so pegylated versions that turns non-fluorescent compound into fluorescent resirufin, and a sulfonated version that hydroxylates coumarin, again creating a fluorescent compound. And so what we ended up doing is very simple, is to put in a microfluidic chip our mammalian cells in one end and the artificial cells in the other end, activating the artificial cells, and took microscopy images. And in the actual samples, we could see that the fluorescent intensity inside of the cells increases compared to the control, suggesting that we actually do have this very simple first step accomplished. But this is obviously very simple. It's just a diffusion of a fluorescent compound. So Isabella, the PhD student at the time, was like, okay, that was fun, but how about we do something a little more advanced? And she asked then the question, can we at least eavesdrop on our artificial cells? Or can we use the artificial cells to eavesdrop on our liver cells? So what we needed to do for that is we needed to have a cleavable signal that is being released from the artificial cells that then interacts with the metabolite from the liver cells, and that we can then detect by our artificial cells again. And so we can then, depending on the signal we're receiving, we would get an idea of how that specific function of the mammalian cells. So what we ended up doing is our artificial cells, when we work with, when we integrate them with mammalian cells, are typically made out of alginate. So we conjugated cysteine to this via disulfide bond, which we can then easily cleave in either enzymatically or with a small molecule that releases then the cysteine. Our cells convert cyanomethoxybenzothiazole into its hydroxylated version, which then reacts in solution, forms luciferin, which we then can read out as light. And so that's how the experimental setup looks like. At the moment, they are still separated. So the artificial cells here floating on a paper chip on top of the mammalian cells, here seen in a 96 well plate setup. And what we're monitoring is the luminescence that comes from the produced luciferin. And you can see if we now, and this eavesdropping really works, because if depending on if we induce or not induce the cells, we get a different signal. And it doesn't matter how we actually cleaving the cysteine originally with the enzyme or the small molecules, the signal outcome is really the same, but the limiting step, or that the step that makes the difference, comes from the mammalian cells. Now, this is clearly what we as chemists or as researchers working in bottom-up synthetic biology are interested in. Can we imitate the key functions of a certain tissue, the P450 enzyme in this case? But when we're more interested in, can we actually be clinically useful, or can we actually act as supportive units? What is much more important are reactive oxygen species in the liver, because too high levels of them is the source of a lot of problems within the liver. So almost 8 years ago now, that's where we actually started. So our very first efforts in 2017, when we decided, okay, what happens if we want to integrate our artificial cells with our liver cells? We actually were looking at reactive oxygen species stress. At that point, very simplistic. So we just took our alginate-based artificial cell, and we added an enzyme to it, catalase at that case, in this case, because that will convert hydrogen peroxide and remove it out of the solution. And by doing so, we were expecting that the cell viability improved, which is essentially what happens. So you can see here, if you have the catalyst containing microreactors, the viability of the cells is much higher compared to the examples where we only have the ghosts. Now, this worked for exactly 24 hours. If we do the same experiment after 48 hours, we will not see any difference anymore, because the artificial cells, they cannot recreate new catalase, which means that the enzyme loses its function when it's in cell culture for extended period of time. So that is one really big problem we had to address. And the second aspect is that while our mammalian cells proliferate, our artificial cells don't. So we need to make sure that from the very beginning, we have a very high amount of artificial cells integrated in our proliferating semi-synthetic tissue. So what we originally did here is we just polylysin coated the alginate, which helped the mammalian cells to integrate, but it had clearly its limitations. So the second aspect we decided to overcome by camouflaging the artificial cells with cell membrane vesicles. So what that essentially means is that we decided to purify the plasma membrane from our HepG2 cells, turn them into vesicles, and stick them around our artificial cells, so that from the outside, the liver cells, that the artificial cell will look like a liver cell, at least on the membrane level. And so that took us quite a while to get this actually to work, to purify the cell membrane vesicle. We preserved E or N coherence, but eventually it worked. So we can use the alginate beads, put PLL on top, and then coat it completely with the cell membrane vesicles, as seen here, with the cell membrane vesicles marked in green. And then if we use an antibody for N coherence, we can also show that we only have signal coming when we do have the cell membrane vesicles present, and not when we just have PLL on our particles. So indicating really, that is a strong indication that we do have the proteins still present, which we wanted to have. But the question is then, do we actually see the calcium-mediated adhesion between the coherence on these artificial cells? And so what we made is beads, red and green beads, just fluorescently labeled. When there is only PLL present, independent if we had calcium or not, nothing happens, they just beads in solution. But when we now have our cell membrane vesicles and we add calcium, what we ended up is making these cute little tissue aggregates that can be suspended again if we chelate the calcium. So what worked with these artificial beads is the question, will it also work with mammalian cells? And so if we only have PLL and we change the ratio between the artificial and the mammalian cells, we have some level of integration, but it's definitely limited. When we have the cell membrane vesicle, it's not much better if we don't have any calcium. But if we add the calcium, it looks like that both cases all of a sudden form a much more coherent tissue. So it's very difficult to judge from these bright field images. But if we then take confocal laser scanning microscopy images of that tissue, when we only have the PLL present, we see the tissue fragments just from transporting out of the well onto this, the glass slide for imaging, while when we have the cell membrane vesicles present, we're really forming a coherent, very thick, very stable tissue that can be transferred with a pipette without any damage, clearly showing that this more nature-inspired way of using the cell membrane vesicles instead of just PLL really improved the integration. Now, the second aspect we wanted to address is how can we improve the catalytic activity. So instead of using an enzyme, we decided to use a small organic compound, so EUK. We did not invent that, that comes from the 70s. But back in the day, most of the commercial compounds, they cannot be chemically modified, and they're also not catalytically very active, but they are imitating the catalase and superoxide dismutase activity. So we synthesized a variation of that compound, leaving some active groups for further chemical modification. And we also saw that our catalysts are actually much more active in removing hydrogen peroxide. For instance, look, comparing here the one with the stars, which is EUKB here, compared to the commercial one that removes hydrogen peroxide, but on a much more moderate level. And so this catalyst is now active. It's not a protein, so it will survive the cell culture without much problem. And so what we did then, we integrated these artificial cells with the cell membrane vesicle, but now they have a catalytic core in addition, or they have ghosts, so they beads without catalytic activity. And then we stress the samples, so we stress them, we increase the ROS levels in the surrounding, and we can see then when we only have the ghosts present that the cells start to die. So red cells are dead and green ones are alive. But when the catalyst is there, we actually can stress them quite high levels, and they stay alive. What is though more interesting when it comes to clinical application is the question, does it actually have an intracellular benefit? And importantly for the liver is, does it actually support the mitochondria? Because the liver tends to have an energy crisis, so the mitochondria is the most important organelle that needs to remain in homeostasis. So I don't want to go into details here. What I just would like to highlight is that the bluish bars, which are the active artificial cells, they don't have any statistical difference between ROS stress or no ROS stress compared to the controls where we have the ghosts. And that is essentially what we want. We do not want to have any change out of the healthy level. So this is actually quite an interesting accomplishment, that we could have our artificial cells providing extracellular support, which then intracellularly helped the living mammalian cells. Now, but these tissues are still very small, so they're like in the millimeter range. So we really wanted to increase the size of the tissues, of the semi-synthetic tissues we can make, and we want to see, can we actually 3D print our artificial cells? And so to do so, we went back to our more cytochrome P450 active enzymes to do that. And so what we did, we created a composite ink that consisted of artificial cells and mammalian cell aggregate. So the mammalian cell aggregate, we're making very simple. We have a non-adhesive flask, and then the HepG2 cells form quite reproducible little aggregates. And then the artificial cell essentially act as a granular material in a liquid phase ink, which really helps on some level the printing properties. But if we have too much of the granular material in it, the printing really becomes very lumpy and not very nice to look at. But what we can see in these prints, when we do electron microscopy or fluorescence microscopy, here in the black holes are essentially the artificial cells. So initially we had very few, and then as we go here, oops, we keep having more and more of those artificial cells present in those prints. Now we combined the beads with the cell aggregates, let them proliferate over 35 days. What we really could see is that the alginate-based beads remain intact and eventually are completely embedded or surrounded by the growing mammalian cells. So in this case, because this is an example with hepatocyte, HepG2 cells, which is a cancerous cell line. So what we're seeing here is essentially little mushrooms growing out because of the cancerous natures of the cells. We have started working with more relevant cell lines like the hepRG cells, which are not a cancerous cell line, and there we do not have this like mushroom outgrowth. But now, what's the most interesting question? Can we actually have an additive effect of our artificial cells in a specific liver-like function within those semi-synthetic prints? And so what we here comparing is the prints where we have ghosts. So the beads are present, but there's no catalysis possible, compared to then when the beads can convert or can create resolution as a fluorescent signal. And so when we do these experiments after 4 days, we can see that we approximately have 6 nanomoles higher readout in the case where we have the catalytically active artificial cells present. So 6 nanomolar probably doesn't sound like much, but it's pretty much exactly the amount we would get when we would print only the artificial cells without the mammalian cells present, which really shows that these small organic catalysts are really very stable. They do not mind the cellular environment. And the same catalysis is then preserved after 14 days. So we keep the 6 nanomole increase. We did the experiments also after 28 days, but the difference is at that point, the mammalian cells have proliferated so much that we didn't see any difference anymore. And so with that, I hope I could give you as a short insight into what we are doing in my research group. I would really like to acknowledge my collaborators, in particular the ones, the clinicians who are working with us, who are prepared to listen to our crazy artificial cells, and then the ERC and the Lundbeck Foundation, who actually funded most of the research I shared with you today. And thank you very much for listening.
Thank you. Thank you so much for a great talk. There is a lot of questions in chat. Can you see chat?
Yes, just give me a second. Yes, hang on.
If you could read a question out loud before answering, because people watching the recording won't see chat.
Okay, so the first question is, what is the composition of the hybrid vesicles? Hang on, I have this weird, I need to stop sharing, I think. Yeah, okay, that's better. Okay, so the first question is, what is the composition of the hybrid vesicles? Yes, they are made out of lipids and copolymer. So it's an amphiphilic block copolymer with cholesterol units in the hydrophobic block and a lot of variations in the hydrophilic one, and then lipids, very often just DOPC lipids. And then the next question is, how do you make the protein made in vitro or purified from bacterial sources? So it's so that's not my expertise. We just get it. So it's expressed in bacteria, and we get it then purified. And what I probably should add here, because it was really quite impressive, that the reconstitution is super easy. We do not even need to add additional surfactant. Just the surfactant that comes originally with the flip is enough to just insert the protein in the membrane. So when you put the magnetic motors inside liposomes, when they move, do they deform the membrane? So when they move, so when they're inside, they get stuck on the membrane and do not do any membrane deformation, because when they are inside, they slow down so much that they don't have enough energy or power to actually do something to the membrane. If the question is more like when they are crossing, so what we're seeing when they're crossing is that they don't punch holes like a sand through a tent, but they really do more like sort of endocytosis kind of thing. So they take pieces of the membrane with when they're crossing. So then on slide 31, how could you put the artificial cells on the upper of mammalian cells? On which container inside the plate? Okay, I need to check what's on my slide 31. Let me see what's on my slide 31. Oof. Okay. So what the way we're doing this is, so we have a paper chip. So this is actually a quite interesting thing we came up a few years ago. So what we're doing is you can use a regular printer to make that paper chip float. You need a hydrophobic barrier, essentially. And so what we're doing, we're using a regular printer to print a ring on two sides of filter paper. We put that in the autoclave, which then makes the polymer particle of our regular printer melt. And at the same time, it sterilizes the material. We can then just cut this paper chip out and put it on cell culture, and it's going to float. It floats for like weeks. It's absolutely non-toxic to the cells, and is essentially our solution to the wax printers, which we couldn't purchase anymore. And so we just had that as on top as the floating material or as the floating chip. So it's essentially a cheap version of a plastic insert in the well plate. Okay, and then when you co-culture artificial cells with natural cells, is it a stupid, do you see artificial cell lysing and releasing any possibly toxic to the natural cells? Well, so, I mean, we cannot say by 100% that they are lysing, but our artificial cells are made out of, so they are hydrogel bead. They're fairly stable. I mean, we are always surprised how stable they are, because technically they should slowly degrade by the calcium exchanging for the sodium, but we never really see that, and much of that, so it really stays stable. We have not had much issues with toxicity, so we're encapsulating in it, and because it's alginate, the cells cannot degrade the artificial cells, so it is preserved. So it will be obviously different if that is made out of gelatin, but with alginate, we do not have toxicity problems that by our artificial cells falling apart. And then does ROS affect your liposome membrane stability at all? So, I should probably be, for clarity, so when we do the work with ROS, which is with the cells, it's always in hydrogel bead. At this point in time, there is no vesicle yet. So I can really not answer that question because I don't know. And so what is the bioprinting resolution? How small of an artificial cell can you make? Okay, so what we do with the bioprinting is we do not make the artificial cells with bioprinting, but what we actually do, we printing the tissue with the bioprinter. We're making the artificial cells beforehand. There was a time when we used microfluidics, but what we have is it's called an encapsulator. So it's a commercial instrument that makes beads, hydrogel beads between, I guess, 70 microns and up. It's not as perfect or as monodisperse as when you make the same beads with microfluidics, but it's just much easier, and the yield is huge. It's super easy to make. So that's why our artificial cells are a little larger than one would probably wish for. And then in co-culture systems, do synthetic cells influence the mechanosensing pathway of natural cells, given that they may have different mechanical properties compared to normal cells? Well, that's a very interesting question. And we actually, when we originally started the project, there was this one task we wanted to do by varying the mechanical properties of the artificial cells, so making them very stiff and then very close to the liver tissue, to see if that actually makes a difference in terms of how well it integrates or how well they proliferate. And on our very superficial analysis we never saw much of a difference. And so we decided at some point we're just going to take we measured a whole lot of different compositions of alginate, and really it does not make much of difference. So we just went with one specific part. So, but we never really looked into this. And the liver cells don't seem to be very sensitive for that compared to other kind of cells. Yes, and I think that were all the questions.
I think so. I think you basically gave a second talk, but that was great. I was having a similar, because we've been trying to co-culture synthetic cells with natural cells too, mostly with bacteria. So I was really curious if you ever looked at any toxicity, because we haven't either, but I have a sneaky suspicion that there's something there that we should be looking at. Like I know some of my synthetic cells did burst when I co-cultured with bacteria, and they release all those guts that are basically TXTL. I think we'll need to, that the royal we of everyone working on it will have to look at it eventually.
Yeah, by all means. I mean, we also have much more simple stuff inside of our artificial cells, right? We have very controlled, like a single catalyst at this point in time, or two catalysts, but nothing like entire cell lysates and all sorts of DNA templates and things. So we have a very simplistic environment. And I do think it's because we have hydrogels, the bursting is not a problem, and the bursting is definitely going to be a problem with the giant vesicles. I mean, our mammalian cells, what they do with the giant vesicles, they really crumble it up and even internalize it afterwards then. So we tried that in, I don't know, 10 years ago and decided, nah, that's not, for now, definitely not the way to go. So yeah, but by all means, there will be. And then I guess bacteria are also different to mammalian cells.
Yeah. So, yeah, my bacteria were actively trying to eat my synthetic cells. You don't have that problem.
Yeah.
Yeah. All right. Thank you so much. That was really cool talk. I want to say thank you and thank everyone for a great discussion, and have a great rest of your Monday.
Thank you very much.
Bye, everyone.
Bye.